Can I start now?
Okay, let me just say a few words. So, good afternoon and welcome all to this SMCB seminar series. So, on behalf of my co-organizers, Professor Ranjit বিশ্বাস from SN Bose Center for Basic Sciences and Professor Rajiv বিশ্বাস from IIT Tirupati, I welcome you all. And so, today it's a very special day because none other than our mentor, Professor Biman Bagchi, is going to give this very special seminar today. So Professor Bagchi has been the mentor for this SMCB seminar series, and it's his brainchild. He decided to talk to us today to bridge the gap between experiment and theory. There are many concepts that many of us often don't realize. So just a few, of course, Professor Bagchi doesn't need any introduction.
Yes. You have to say it enough.
Yeah. Yeah. So, okay. So I request Professor Bagchi to please take over.
Thank you. Yeah, it is always, I agree with someone that is always good to have a formal structure, because once Peter Walliness was giving a lecture somewhere and he was, everybody told, okay, he's a guy who knows everybody and all this, we know him and all that, and he needs no introduction. He was so upset. He later told me that is not correct. They should say at least one or two words. And same thing I have seen they did with David Nelson in Rise. They also told him he does not need. And he was also not too happy, because if I am talking of Nelson, I would certainly talk of his two-dimensional melting, right? And so many beautiful work that he has done. Okay, coming back. So it was, I gave a talk in NCL, some McBain memorial lecture. I was very pleased to get that, because I didn't know, unfortunately, I've removed the slides of McBain, that McBain is the person who coined the name myself. And he's also very big in colloids. He retired from Stanford. Then Santisharu Bhatnagar somehow managed to get him here, which is must be amazing that the retired man come when there's nothing, when NCL started, nothing, they're just rocks. They showed some pictures. And there, Suman probably knows the details. Then in 3-4 years, he built a beautiful building. I think he came in 47, left in 52, something like that. And how he got the money, how he got the permissions, all these things, the mystery. But he was a very famous scientist. I gave that lecture, and it was very nice. I didn't know. McBain, I knew the name when we did work on myself, but I didn't know he was the founding director of NCL. So the talk I gave, which was well attended, and even Sarika said it was a nice talk. And so I hope Sarika is not here. This Suman, you edit some of my jokes. You please, my students edit them with all the things I say even in my lecture. Now coming back, that this is something after giving, and also I started thinking that for students, we should do a little bit more fundamental level, little bit more simple level talks, and bring them a little bit in the history. That's very, very important. Why? Because if you want to understand the most modern techniques that are coming, both theoreticians and experimentalists, like time domain X-ray, all of us get a lot of information and letters about postdoc or PhD positions. Now, huge number of them are in Europe, and using time domain X-ray and many different techniques, single molecule spectroscopy and many higher. Attosecond is a big thing. But we need to have a certain idea of what is going on, how we came to that, when did it start. If you have certain historical perspective, that really will help. Okay. So, so these are the book that will be important for today's talk. Is this the non-equilibrium StatMech book, which just came out a few months ago, and now is available. They have done a paperback of the first one, and they want to do a paperback of the second one, but probably they will wait a little bit. Okay, so now this is something I every time, it's very, very nice that a little historical thing is there. And that man always have talked of time and how to measure time. Then there are all the clocks which are based on sun. Then we looked at the stars. And of course, we needed to know the flows of rivers, tide and ebb, and all these things were very important. And these must have come more than 10, 15,000 years ago. In ancient India, a little later, 4,000-5,000 maybe, we had the Sonatoni. Unit is a wink of an eye. It's very interesting. Now, what is at stake is the information contained in time correlation functions. So, in modern days, when you look into information from an experiment, what is that people try to measure? They measure the line shape. Now, line shape, optical line shape, or vibrational line shape, or rotational line shape, they are all infrared or Raman. They are all Fourier transforms of time correlation function. I'm not going to talk on that because I'll talk more of phenomena, but I want to emphasize that, and I'll come back to it again. So you have a time correlation function like in vibrational isotopic Raman line width. You have the normal coordinate q, which we know, like in oxygen to O, distance is the normal coordinate. Water has 3 normal coordinates: bending, symmetric stretching, and asymmetric stretching. So what you observe experimentally are the line shape of these normal coordinates. This is one of the fantastic understanding came from quantum mechanics. However, when you do at the line shape and want to do the decay of that or interaction with the surrounding media that a physical chemist or a chemical physicist wants, then you need to know the line shape is given by this. Q is the normal coordinate, the Q0, Qt, Fourier transform of that, one-sided Fourier transform of that is the line shape. Similarly, infrared, it is dipole moment and mixed with a little bit of normal coordinate. NMR, rotation correlation function. So we have all, whatever measurements we do in lab. When I first joined SSU, I was finding they are doing XAP and all the other things, and they are just getting the position. So I used to bug them, Professor Hegde and other people, that look, you guys are getting okay electron photoionization 531 electron volt. I said, but you guys are getting a line. Why you are getting a line? It should be, if you look very carefully, it will be little breadth will be there, because you are doing in the, not in the gas phase, you are doing in a surface, and you are ionizing core electrons, that photoelectron does. But I didn't get any answer. I hope they do a little bit now. Probably their characterization, they are important. Now, there are response functions, just like shape, size, and system properties. Also have viscosity, thermal diffusion coefficient, translation, rotational diffusion. It all comes from the spectroscopic measurements. And spectroscopic measurements interface with this theory. So that's the reason of this title of the talk, relation between theory and experiment. So I'll talk a bunch of experiments and I'll talk a bunch of theories. But underlying is that I want to students to know and young faculty to know that there is a very interesting relation. Many times you can make a very fundamental contribution if you go to an experimentalist, and then if you can get a result from the experimentalist which is to be explained. Sometimes that's how many big discoveries are made. Okay, so but we needed time domain, but time domain was not available till 1980s, late 80s actually. So before that, only frequency domain measurements are available. I was saying for spectroscopy, NMR, Raman, and all these things. And neutron scattering, of course. Now, how these frequency domain measurements were useful in understanding the system. As I'm saying that they are either thermodynamic response functions like specific heat, compressibility, dielectric constant, or you have dynamic response functions. And dynamic response functions are viscosity, conductivity, these are. Now, basic philosophy is that in order to know about a system, whether it's thermodynamic or dynamic, you need to perturb it. You have to push it around. Just like you want to know the mass of a person, you have to push it. Essentially studying the response function, but in these techniques, we are getting the response function not thermodynamic but microscopic. So when you perturb that, what do? Frequency domain measurements, all are exchanging energy and momentum. Okay, neutron scattering is momentum, and these optical measurements are all energy. So frequency wave spectrum, which is just a second, let me minimize. Okay, which is the spectrum of the intensity versus frequency. Example of Rayleigh-Benoist spectrum, the big thing, and Rayleigh-Benoist spectrum described in my book at detail is the light scattering, optical light, 5,000-6,000 Armstrong wavelength, that gets scattered. Why it gets scattered is a beautiful story. But they gives you, these are frequency domain measurements, they give you what? They give you bulk viscosity, they give you thermal diffusivity, they give you velocity of sound. They get an enormous number of sound attenuation coefficient, which in turn shear viscosity and bulk viscosity. So Rayleigh-Benoist spectra give you 5, and one of the very rare information. So, however, many times from the spectrum, when you want to go time domain, then what we get is not very reliable. Why? Because there's this problem of the information lost on the winds. So because short time is high frequency, and when you do the Fourier back transform to get the time correlation function or the short time decay, we don't get it accurately. This was a huge problem, and a huge problem which continued. That's why I remember that our Graham Fleming used to give the talk and always used to show a spectrum, which I'll show also, which is a big block. I remember he used to talk iodine in ethanol, and in the big blob, he said, okay, this has no meaning because, rate is the width of the line shape. This certainly cannot be any rate. So we want to know what is inside. So the term that came into is inhomogeneous broadening. That means your line shape that you are seeing in frequency domain is not the one transition. There are many transitions mixed together, and all the information get lost. So, so in homogeneity, all experimentalists go. There are those who are spectroscopists. Don't look at the left and right. Look at the middle. This is what observed spectrum is the middle one. This is a huge spectrum, and as I said, rate is the decay of the time correlation function is the width. Usually we do full width at half maximum, and that is now you see if it is a sum of many homogeneous lines, each are very narrow, then you can be, if you don't know, then you will land in complete trouble. So frequency domain measurements ran into this huge travel from people all over the air. That is the time I think laser came in, and people started now interrogating by tuning frequency of the laser, and which of course not those days greatly tunable, so they selected system, but they could study the one Lorentzian line shape. Okay, and so time domain, I quickly wrote, this is the one I think made by Rajiv for KBPI many years ago, and is I like it because of the horse. So how did it start? Actually, it started side by side in Europe, which I usually don't cover. I should probably do that. So it is this famous bet between Stanford, it was in Palo Alto in Stanford University. I think he was the mayor of Stanford, Palo Alto then, and a very rich businessman. And they, in one of their drinking bout, they argued whether the 4 legs of the horse is up in the air. So one of them said yes, another said no. How do they say? So they looked into racing horse. So it was the race, because they are big race course guys. So they looked into, they had all the horses themselves, and they couldn't figure out. Then they hired the master photographer, Edward Maybridge, who did this time delay experiments. I think some of you might not have heard story from me. So he had 16 of the camera lined up, and then all of them now connected to one which is going to pull string by different lengths. So when he pulls, then one by one, the shutters go up in a split second. So he could generate 16 snaps. Now, in Walt Disney, these animation movies, in our time, there used to be 32 snaps in 1 second. So that's a lot of work, you know. Now here, then he put it. This is this original movie put together with these 16 snaps. And yes, you can see in one place there, all the 4 legs indeed up in the air. So this was the beginning of, essentially, that I tend to think that might be the reason. Palo Alto, Hollywood is not too far, right? And so movie camera, but also it was going on in Europe. Someday I'll talk about the Europe, and what was going on in Europe, because there are a lot of fantastic people in Europe those days, you know, Renaissance, post-Renaissance. Okay. So, however, the reactions that a tried to do is the I used to have one Manfred Eigen thing which is not here, so probably it will come later. The Manfred Eigen thing will probably got deleted. So fast chemical reactions of the 70s or 80s were done by late 60s actually by Manfred Eigen, and Manfred Eigen did the and Norris and Porter. I think Norris and Porter did the flash photolysis, and Manfred Eigen did the temperature jump method, or other way around. And they could first time measure in microsecond. Before that, if you look at all the books, I remember we had this Finner or whatever these books, their old book, and Finner was giving many reactions, and they're saying, well, the rate is not known, it is too fast to measure. So that is what actually started Manfred Eigen. He started to, he was intrigued in in his classes in 1940s or so, that when teachers say these reactions are too fast to measure. So then he went, the undergraduate class, he went ahead to measure that, and also did Norris and Porter. Now, I would like you to point out that here, there's a beautiful novel lecture by these people, you know, which you should really, the novel lectures are just wonderful. Okay. Then came the laser spectroscopy. So these guys did the gas phase spectroscopy. The not all, the liquid, but they were using really non-optical methods. But this was the flash photolysis and all these things, their detector. But this laser, you know, was given by the Bloombergen and Shaklo and another person in UCLA, I forgot the name. They created, was a very great friend of Amit Zuel, because he used to come now and then, who actually also nominated Amit Zuel. Now, they did the laser. Then, in late 1960s, this was the experiment done. The Nobel Prize was given later by Whitey Lee, Dudley Hershberg, and Polanyi was the experimentalist. Dudley Hershberg did a great Nobel lecture in 1986. Please read that for everybody. Dudley Hershberg's Nobel lecture is a wonderful thing. You know, he was a wonderful person, you know, very, very interesting person, very nice person. I remember when I went to Chicago, my Julian Gibbs's ideas were not that great. My advisor, we didn't have a single paper, and I was very worried. So I walked around and found a polymer, some things in polymer, and I wrote a single author paper, which I sent it out, Journal of Physical Chemistry. It was a short paper. So there was one referee said, yes, accept it, just written on the A. And that was Farid Abraham. Other guy said, okay, there are these extra things to be done. And Dudley Hershberg wrote, it's really beautiful. He said, Biman, I never knew him. He didn't know me, Biman. Since this is your first paper, I'm going to accept it, but correct this English. And he corrected the English himself. Okay. But this is the kind of people who are money, you know, amazing people. Okay. Now, but read he's also very interesting man. He describes how laser came in, how the nanosecond laser setup was done with molecular beam, with Ian Lee and all the time constant, and also how it agrees exactly with the potential energy surface that was done by Polanyi and other people. And then voice through time continued lot in electron transfer reaction, which he did not do the time domain immediately, but he did later. But I'll talk about little bit of his work. And then, of course, the Amidswell, and this is a book. This is a beautiful book. If you can, of course, these days you will get it free probably. It is a very nice, a little bit more personal account, but still nice to read. Now, what did Amidswell did? When you have a very short pulse, what do you do? Let us think about it a little bit. I need to have a probe. Okay, that can be a camera, like in my average experiments. It can be a couple of pulses. Okay, and now see, optical pulses are the probe, always go with pump and probe. Okay, now say you want to identify. That's what he got the Nobel Prize for, the transition state, and it was femtosecond, maybe 50 femtosecond or so. So he gave this example. There's a cat which is on a very nicely and not suspicious at all, very sitting on a and you just push it. The cat falls. If it is a little bit higher, then what interesting thing happens? Cat always lands on its own legs. But in that, it undergoes an evolution, and these are the synapses. So final state is this, say, almost the same as the initial state on the ground. But there is this state then in middle, which is the transition state. So this is a good example. If I can snap, I know the transition state, but in the real transition state, can I get it? And that's what Amidswell did. But again, palm, probe, molecular beam, all this together. So evolution through time, spectroscopy, as we all know, it started in microsecond, millisecond, microsecond, then went to a Northeast Porter, again, they were in few tens of microsecond. And then Dudley Herzbach and Ian Lee were in nanosecond. Then nobody, there are a lot of work done on picosecond, but one didn't give a Nobel Prize to picosecond, God knows why. Then we thought that they were given one, and then comes femtoseconds, where they gave for because detecting the transitions. Actually, Amidswell did a lot of very nice work, which not that known because it was very involved work. I particularly want to talk a little bit about the attosecond. AS is 10 to the power minus 18 second. So at our time, people always used to ask, what chemistry could be there in femtosecond? Before that, they asked what chemistry would be there in picosecond. Now, I had a lot of time. What chemistry could be there in attosecond? That's a very interesting question. What actually happens, is the last red line first, that attosecond allows you to study electron dynamics, like study of bond breaking or electronic study, illustration of electron. And that, because the electron is so light, motion of electron takes place in probably few tenths of attosecond. That's why now there are a lot of experiments being done. Nobel Prize probably last year, or before last year, came to electron diffraction. And there is one more waiting. Single molecule has been given, and we are waiting for the not given the time domain X-ray. That I'll talk a little bit. Okay. So rates of complex system, in a huge amount of work done. First time, made in year 30. So what I'll do now, I'll talk of some experiments, and I'll do some simple theories, and then I'll go to more complicated things. Okay. However, so long I've talked of dynamics, and evolution of time scales by these people, from millisecond to microsecond to picosecond, nanosecond to picosecond and femtosecond. Now, however, unfortunate part about laser is that laser really does not provide you information about structure. Not only that, many times in complex system, laser cannot give you even the potential at surface. We write something, but that is in a big system not being supported by quantum calculation. So we really kind of dream, we kind of conjecture. And that was actually Amitzwell's thing, that we talk of the intermediates, we talk of transition state, but nobody has seen the transition state. Similarly, many things we write of isomerization, C-strand isomerization, a molecule still being, for example, phenyl rings moving. We don't see that, no? We have no understanding of that. We have optically, we excite, it comes down, we say, okay, as it coming down, it is relaxing. Please understand that laser is very indirect. We evolved from frequency domain to time domain. Yes, we got many things that worked out somewhat better in neutron scattering, but in our chemical dynamics and biology, we still have a problem that we can trust interpretation only for small molecules. Probably they are correct. That's what I have this Dudley Herzberg Nobel lecture. Please read that. Okay. Now, structure determines dynamics. That's the standard thing. Now, how do I get structure? One is microscopy, evolution towards electron microscopy, and really now Armstrong lens scale, X-ray diffraction. This is something I'll talk about it today a little bit. It's extremely new technique, very difficult technique. This is only done. You need the synchrotron, the high intensity X-ray from synchrotron. Tough job, but I'll talk about it. Okay, so now voice through length. I did the voice through time. Let me quickly go through the voice through length coming from arrow should be left to right, but okay, it coming from big our ordinary understanding of matter to subatomic level, where you have electron and a. But in chemistry, I believe we won't go to sub-electron work or all those things. I think we will stop in neutron, electron, proton, at least in my lifetime. Okay. Now, voice through length has also an interesting history. One is Aristotle, who started first time put these relative positions and the length scale. So that's what adapted by Bible and many things. That Earth is the center of the universe, right? The blue one in the center. And then you have Moon. Then you have Venus. And interestingly, Sun is a little away, but he has a big size of Sun. This is Aristotle's universe. Then people developed microscope, lens, and the significant was Zacharias Janssen. First compound means multiple lenses, which could get a depth of, like in many cameras, you have two lenses. And then, of course, these days they have program to take care of that. Then this person, Anton Lichok, who was the person he used, make and use. Those days, you use your own, you don't get it. Study bacteria for the first time, but dead bacteria. But internet, you can find a lot more about his work. Now, very quickly, we got developed, but look at the lengths on a time scales. These were in post-Renaissance period. And then almost nothing happened for 200-300 years. Now, we study objects below wavelength of light. It's still ultra microscope and optical microscopes with the use of lens and light. He could do, and lenses. He could study below wavelength of light. That was great. And this guy is amazing, a genius, Fritz Janik. He could a phase contrast microscope. He could study within the living cells. What phase contrast he did actually, he sent one light inside the cell, living cell, and the other side he sent the so one goes in the cell and one goes outside. Then he subtract the two, and that is in the liquid, of course. And he could get what is the thing of the this phase contrast microscope. Then he sees, the microscope. I used to know a little bit more, but I have not read up recently. Now, first design of the first electron microscope was done by Raska, and the Nobel Prize. These were done by Rajiv many years ago. And these got Binning was our time, they started doing. Then came all kinds of scattering, scanning, tunneling, SPM, all other departments of electron microscopes that came in. And all were got Nobel Prize. So we get structure now. However, this structure everybody gets, and hugely important in nano, has become, because people publish so many papers in nano by giving the electron TEM and all the tunneling electron microscopes and others. And that's a great pictures you see, but that has no dynamics. That's one huge problem. So what do we need? We need to know how a structure changes, particularly in chemistry and biology. How a structure changes, how do we find that? We cannot do by microscope, neither do I get by laser. So there are two avenues that are developed. One is single molecule spectroscopy that I'll briefly tell, and is ultra-fast X-ray spectroscopy. That's a wonderful technique, extremely difficult technique, as I'm saying. See, it is an absorption spectroscopy by and large. X-ray absorption, not X-ray diffraction. And X-ray absorption is very much influenced by our laser absorption experiments. But there is some beautiful thing that happened which allows it to probe the structure. Now, difference in ultra-fast X-ray spectroscopy is that you get a local structure, but you don't get anything. So you get the first neighbor, more or less, maybe second neighbor. That's it. Unlike X-ray diffraction, where you get the glow-Bragg spectrum and the Bragg diffraction, all these things. Okay, so this was the I believe Moener's figure that where he has a tetrazine on a paraffin wax surface, and these lights were emitting are these tetrazine or pentazine. Their reason why they are chosen like that, most of the time why they're excited and that they emit and they don't lose any photon, because we have to detect every single photon. But what is so beautiful, for the first time, that's why people are so excited, that the first time you get you see a molecule, you really see a molecule. Not only that, you see a molecule which is moving, like those broad ones are the molecules, they are moving. This one on the middle and one on the left, some are very, very sharp. Okay. So now, as Francis Crick famously said, that if you want to understand function, study structure. Structure determines dynamics. But how do I determine structural change, both structure and dynamics at the same time? That is the need of the time. That's what I think a lot of even theory is needed. I remember I started doing, one of the reasons I was invited by Sunny Z was that I started doing single molecule spectroscopy theories. I did an average job. I did not do a great job. But yes, those are the first days. So X-ray absorption, in contrast to diffraction, is a disordered system. You get liquids, glass, and hopefully isomerization. You need a very intense X-ray, and those intense X-ray you get from that, not your tabletop. But I'm hearing that they are already have a high intensity in tabletop, but you get to synchrotron. Okay, I'll talk a little bit more. So the experiments and theory, rapid growth in both after 1960s. I'll talk a little back, look back a little, and then come back to single molecule and X-ray diffraction. Okay, impact of short-lived chemical reaction dynamics is huge, huge. That was whole 90s, 80s, and 90s. Isolation reaction dyes, malachite green, crystal violet, Stilbene, solution dynamics, barrier-less chemical reactions, trans-cis isomerization in Stilbene. When I first came across these things, I was like this. David Oxford was on sabbatical. I was so happy. I was moving around. I came to my mini sabbatical myself, spent 2 months. And here, happily roaming around. And when I went back, people were not greatly happy. David Oxford once came and told, go and talk with Graham Fleming. He has some interesting results. Okay. And then Stuart Rice got hold of me. Biman, what are you doing? I was doing nothing, so I couldn't tell anything. But he said, come and lunch with me. He took me out for lunch and gave me a problem. Somehow, the word went in Chicago that Biman is enjoying. and so they may try to stop my enjoyment. Now, but when I went to meet Graham, David sent me to meet Graham. He had this beautiful breakdown of Cramer's theory results. The whole, everybody was talking. He was giving, Graham told me, I'm so tired of giving talks. Every week, they are asking him from every place to give talks. Okay. So Graham tried to explain a little bit of experiments to me. I didn't understand. I still remember he says that I feel very sad when I have to talk of experiments to obstreperous people. So that really insulting. Okay. But then, then A told me, very interesting. A told me that David told me there is a theory by Hines. Okay, I'll come back to that. That is a really nice story. But if that was done, let me tell the other story. This story is amazing. When the isomorphic breakdown of Cramer's theory I did, then those by the name Graham Fleming and those people used to call their home for tea and all these things. He called and told me, Biman, there is this barrierless chemical reaction. There is no theory, or there is a theory by Forster and Hoffman, two great people, Forster and Hoffman. They did a theory which does not explain the experiment at all. It's e to the minus TQ. So I wanted to get rid of the whole this quickly. So in the board I knew all the probability theory. That's what I did in my PhD days. So I solved it by method of images. I said, this is the solution. I'll show you the solution. He was stunned. Now, these barrierless chemical reactions are ultra fast. Graham wanted to measure them because lasers have come. Surprisingly, they have a rate and a very strong viscosity dependence of quantum field. Many things have been done. Crystal violet, malachite green, and all ethyl violet. They are this kind of TPM dyes, this crystal violet. And what actually happens that it rotates, and the theory was that it comes down. Okay, so this is what, but there are a lot of very interesting things. Here is that you excite from the left well, then there are some relaxation go on, and then it comes down. Now, it stays in excited for some time. How do you find that it isn't excited? You do a very famous technique called excited state absorption. Excited state of ESA, excited state absorption. You excite on top of that. That's some arrows that show there. And then you study the fluorescence. What happens? The fluorescence disappears because it comes and there's a sink here. And you study the fluorescence decay. Now you can also do the ground state recovery when it comes back here. So what one found that decay of excited state absorption was the fastest. Second was the fluorescence decay, and third was the ground state recovery, which are actually said here. So up conversion, 0.1 is shortest, and then the transient absorption or fluorescence decay, that would be 0.2 picosecond, and somewhere, or transient absorption, I think this one. And then somewhere ground state recovery would be some picosecond. Okay. But here are three processes going on. Very interesting. So here is, I took from conical intersection, but it actually works here, that you excite here and a blue upper surface, and it can now execute a two-dimensional motion. And it's very, very important because two-dimensional motion sometimes facilitate the nuclear coupling. So it becomes a reactive code. And it is the same thing that I said. They're coming down, you excite, coming down and emit, and it comes here and goes down. What is the model? How to do the model? It's very interesting. What I did was, actually, I put it in the beginning, in the middle. There is, I put a reflecting barrier on the left. I start a population somewhere here, and a reflector is, these are the figures I got from internet. So they are drawn there according to you, but it's a very widely used theory now. I didn't know it is how widely used, because when I was cutting and pasting, I realized there's so many, not a single of my figure is there, but there are other thousands figures are there. Okay. So you have a reflecting boundary condition, you have an absorbing boundary condition, and you can solve it by considerable method of images. And then if you do not have a sink, absorbing sink, you can have a Gaussian sink with certain finite rate. That has to be solved numerically. But here is the solution, the analytical solution, which I did on the board. So B is A is the diffusion constant, kVt by zeta. B is mu omega square by zeta, those days by notation. And x0 is the initial position, and x is the, so now, because of this guy comes to equilibrium, to minima. And that is how it is coming from, starting from here. Initially, it is very sharp, then gets broad, and it becomes asymmetric. That is the reason it gives to a lot of interesting features that experimentalists knew about it. Okay, one of them is I don't have, but one of them is the blue edge recovery. That means you get a decay in the blue edge, which is the high frequency, and you have a growth in the rate, which is the near the minima. So that was the signature of something very interesting happening. Okay, I'll have a graph of that. Okay, so this is the barrierless chemical reaction. Before that, however, we did activate. It was really not just me. A lot of people are working on that. A lot of fun time there. So Graham's this experiment showed that breakdown of Cramer's theory. So Smolkovsky Cramer's theory is the black line, and red is the near saturation. It becomes very, very weak viscosity dependence. Eta to the power minus 0.2, 0.3, and all these things. Graham found something very interesting. That exponent of viscosity depends on the solvent. So he conjectured that the solvent is controlling the barrier, and hence the barrier height. And that was proven later. But we need a theory then. Cramer's theory completely breaks down. And in a high viscosity, it is essentially Smolkovsky. Cramer's goes to Smolkovsky, and it's very bad. So then, how do you explain that? That I was saying that the idea was that in all these reactions, transition state barrier is a very sharp barrier. That is a barrier frequency omega b. If omega b is large, then the particle doesn't stay there for too long. If particle doesn't stay there for too long, then it does not explode low frequency motions. But these low frequency motions are the ones that determine viscosity. So my reaction gets decoupled from viscosity. So you have a residence time, tau is 1 over, this is small and omega is large. And many cases it is 10 to the 13-second inverse. And that, at that high frequency, there are only very high frequency response. Liquid, if you hit it hard, then liquidity becomes a viscoelastic. It recoils, but do it very hard, you won't be able to go in because it's elastic response. So this is the viscoelasticity. Those days became pretty hot. So what I did, I could use the Cauchy-Heinz theory, which is given here, a self-consistent relation, and self-consistent relation of reactive frequency lambda r, which gives the rate, and omega is the barrier frequency, and frequency dependent friction. Look at that, it depends on this frequency. And I got many things. Ranjit also worked on that. We got the Zwanzig friction, and we got this beautiful, very close relation. Actually, later, this guy, Sumi and people did some experiments, high pressure experiments. They again did wrong. And that is the one Ranjit corrected, because at a very high pressure, you have a residual friction. Your viscosity is in many pascals. And you think, okay, it will decouple completely. No. There is one part that is short-term, short location, short distance repulsive interaction. And so this is here the viscosity dependence in the frequency. And this is the right-hand side is translated into that one. So when the solvent is there, now Cauchy-Heinz theory is huge. Many things, people think he should get a Nobel Prize for this. I have seen in at least 3 international conferences, Heinz giving talk, and they talk at length of the Cauchy-Heinz theory. Dick Zare always said that in India, particularly in India, you chemists don't celebrate the success of your colleagues. I wrote a blog on that. We cannot praise ourselves. But physicists are so good. You know, the work that Sriram Ramaswamy did was good, but main work was done before, like Viksek and Toner, two Toner. But what he did was very good and remarkable. But the amount of credit he gets in India is amazing. You know. Now, in India, we don't, but Heinz, I saw abroad internationally, huge amount of recognition he has. Well, he has done many great work, but Godian stand out. So Godian's the high frequency, one needs to consider frequency dependent, which means you have to have a non-Markovian rate. I'm not going to go into that. So frequency dependent friction and barrier frequency, as I said, these are coupled here. Frequency dependent friction here and barrier frequency there. Self-constant mu is the effective mass. And a frequency dependent friction comes, you can calculate many ways. We have done from Zwanzig friction, first one, then mode coupling theory with Ronjit did, then somebody else did it also. Okay. Now, so I really missing one beautiful slide which I had for the this blue age decay and red age rise. That's really beautiful. Okay. Now we'll go. I might have one later. I don't know. I usually many of the slides are made in yesterday and today. Okay. Now the electron transfer reaction. This is something people don't fully understand. That the reaction coordinate. Why he got the Nobel Prize? He got the reaction coordinate is a collective code. See, till then, before these things, all the reactions were okay. All the reactions were bond breaking kind of thing, but and it was even before isomization. So people did not really know how to talk about outer sphere electron transfer reaction. There's a model by Libby on the right side, which was wrong. He has a radiation list, but A did Rudy Marcus. You know, I took his class, and he was a very interesting man. But I don't have time to talk about his stories. But he realized one thing, that solvent must be halfway between donor and acceptor, electron in donor and electron in acceptor. He must change. And only when it changes, that's called non-equilibrium polarization. And he thermodynamics of that by doing Maxwell's equations and things like that. It's very interesting. So this solvent has to change. Otherwise, electron transfer would not take place. And that is on the shade in the right side. So Marcus reaction coordinate is solvation energy. So we need to know how fast is solvation. This was the biggest story in 1990s and also 2000, I think 2008 or 10. So here one is done that you look at the x coordinate, this is the solvation coordinate. So as I move along the solvation coordinate on the right, energy comes down. So first I excited with the blue line, typically Ramazwell's drawing in front of me did, and then it is emitting. Okay. So, and you are getting shift. Now comes the theory. Okay, when my second work was done, the barrierless chemical reaction, Graham said, Biman, there is no theory of solvation dynamics. We need to do some experiment. There are one or two theories which are wrong. One of them was by Yoshihara. Theory of equation. He connected, he tried to explain by tau d divided luxation time. So I again went and did that. These were done actually 3-4 days. How I did it, I tell my student, the stupid you see in front of me is not. I had one day I also used to be smart. So here is the equation. But this is the first time the longitudinal luxation time came. The appearance of longitudinal luxation time, but the first time that I'm not doing. But that explained huge number of now I have the one. Yeah, this is decay. Look at that. If a solvation takes place, then in the blue age, which is here, you will get a decay. But in a red age, you will get a rise because population will move in. This was Graham's idea. I then did not appreciate what exactly he's doing, but this was his view and is needed. And this was The beautiful, beautiful, and that is the signature of solvation dynamics. And Graham then went ahead to do the water of 100 femtosecond or so, the beautiful work. Actually, he should have gotten Nobel Prize. We moved into biological water, and this again an experiment on the left-hand side. So everywhere, these guys are experimentalists. And Graham Fleming, Amit Zuel, this is Amit Zuel. I should have put his picture here. Now, there is, I should have got Ethik and Grant here also. Utrich is there. They're not, all these are experimentalists. They had this picture, the dialectic laws of a protein, aqueous protein solution are three part. One is very fast, that is liquid. One is very slow, that is protein, as I was showing here, protein rotation. I have all picked up from internet. But there is in between a crossover which is 50 picosecond, which is called delta dispersion. So I believe it was Nilashish who first brought it to me. And then, of course, Amit Zuel, Konkon got very, very interested in that. So we built a model. It was fun trying to calculate the hydrogen bond lifetimes. And I said that, okay, the emergence of slow time is predicted due to stronger. And that we did some theory, very nice theory. Now, please, and yeah, experiments do need theory. And please talk with theoreticians. And theoreticians, please talk with experimentalists. Otherwise, nothing will happen. Even at low level, some interesting things can come out. So different experiments were done and all gave different results, and there are a lot of fighting. Utrich said 300 picosecond. Bartil said no, 4 picosecond. Konkon said no, 500 picosecond. Bartil was extremely, he was so angry that when he was coming here and he saw Konkon is giving a talk, he cancelled his talk in Bangalore. Everything was giving different. QNS giving different. What was the story? This is the story. I used to put Konkon on the top of the elephant. I have seen this many times, that blind men going to look at the eyes of, this is Shomox or Sayantan, I know who do that. Poor elephant that is blind. So this time, what the experimentalists were all partly blind. And somebody was saying it's a wall, it's very slow. Somebody saying, no, it's a tail. Got the tails, it's a rope, it is very fast. Okay. And then this was solved by looking at the distribution of relaxation times, and there's a log normal. Beautiful relaxation time. Every protein we saw, we didn't expect that. And one good thing about that, after this paper published, not a single paper has come on these things. People kind of accepted it. So I was very annoyed at that time that they were so criticizing us. Okay. So it's a long tail, which Konkon got. And then average, the Bartil held, yes, 4 or 5 picosecond, because backbone of water bonded to hydrogen to backbone of protein are very fast. So when you take the average, it is just it. And this is something experiment must be done. I know Konkon told me, Amit Zuel did some, but they're optical experiments. Some kind of a structural thing, some X-ray has been done. Selinger did it, but it's to be done. That how long the protein influence in water stays, that has not been fully done, not resolved. That's what I showed you, otherwise no result. Other thing that has not been done is this Puja's work, that we found some very interesting long range interaction between positive ions and negative ions, fairly long range. And I think there are some experimental evidence in some X-ray kind of a thing, but that needs to be studied. Both theory, Puja lab, theory and experiment, and highly anisotropic in plus minus. These are beautiful things, and these are physical chemistry problems. I think I'll just talk a little and stop. So because I want to talk particularly X-ray diffraction. So single molecule spectroscopy, as I said before. So the famous quoting by Shimon Weiss, the ability to analyze individual molecules still amazes even the most, is the old one that was time old science issue was the nature also did to single molecule spectroscopy. When I first time saw 1999 in Paul Barber's lab, I was just stunned. Also, Biman, these are from single molecules. That is a bullshit. You cannot see molecules. Yeah. Okay, so he said, oh, you are so old-fashioned. We are seeing molecules. Okay. So two things are important here is that ensemble has to be very diluted. But once you dilute, your signal goes down, right? So you need a single photon counting. You need a microscope. So what do? You select by a microscope domain, confocal microscope, whatever, I forgot all this. And then you wait and fluorescence. You keep a laser light continuously on. Then what happens? When a protein moves in, it starts fluoresce, and that you can detect. And then here is shown the same thing, that you big blob is the ensemble, and everyone is a so ensemble goes so wrong. So again, the same thing, partial observation, and this is blinking. I am not going to go. I'll just say one explain that. The same Manfred again. Manfred again did this work, and he said, okay, it takes this much time for a protein to go to a specific binding site. And he did this sliding motion protein, and then it didn't work out. So Hippel and Berg did it. Okay, it also hops. So, these days people are not smoking, but in Calcutta, you don't have to smoke, because in Calcutta, it will happen to you anyway, that your guanine is getting oxidized to oxoguanine. But still, you are not dying because of those of us who smoked. We did survive because we have a wonderful system in the body. HOG1 is replaces and there's an lag replacer. There are all these enzymes which come and correct your mutation. And that is a wonderful thing. Okay. And this is what these experiments, I'm telling you, all this work were done essentially with experimentalists. And in India, it is really difficult to do great work, because if you don't have experimentalists. Here, microfluidic, his test of a DNA, put this protein and polymers, and they are sliding. And he give fluorescence from below and detect from the front, and he knows the mean square displacement. And mean square displacement, pH dependence, everything. So when I went there, they already had the data, but it was not fitting. So there was this theory which was say, okay, it is rotating and translating, but randomly. I said, no, it might as well go along a chain. I remember that it was 11 o'clock, typical sun is meeting in the night. That then I took the tennis. He has a tennis ball and a stick. I told him it might be going like that. And then is a helix. So I could solve that and get this equation, which refers to as Bagchi equation. This is a beautiful equation, which has translational diffusion plus rotational, but then one extra term, which is the next last term here, which is peculiar combination of rotation and translation. And eta is viscosity, r is the radius, rho is the distance of the protein from the center of the DNA, and BP is base pair, pi is pi. And this is what the existing Nidley model does. And this is what our equation does. It's very nice. It was accepted in nature, but then they called and told, see, we tried to sneak in Atlas-Zavos special issue. Sunny told me, Biman, I am nothing, but we have this theory, this derivation of this equation. Why don't we publish it here? I said, okay, I have to also give a paper. I gave that. But now they called, editor called, said, no, you have already published the theory, one half of the result. Okay. Then we do this was what a great by experiments by our Paul Barbara. And there's again a very interesting, when I go in there, I had an accident with a cycle and I was really in pain. I remember one day Paul Barbara gave me all these, Biman, it's like this. These are the results, these are the radius of coagulation and all this. So I calculated and I said, it cannot be fluoric collapse polymer. All of you use fluoric collapse polymer. It cannot be like that, because then you have to bend at every four monomers, MEHPPV. But we know it is at least 7 to 10. So he was really absolutely thrilled and did simulations and everything and found out that this different thing, like random coil is not correct, molten globule is not correct, toroid is not correct, rod also doesn't work, but what works are essentially defects on the last one. Okay, so the last one is time to an X-ray, because I think Ranjit wanted to know a little bit about it. So as I said, if you want to understand function, you need to structure. But how to determine structure and structural change? I've given a lot of references. One is in YouTube. I don't know why YouTube or B. I don't know. There are a lot of very nice YouTube videos. Okay, so what we really want in chemical reaction or in aging in glass or in liquid dynamics, like the one I want to study one day, the ions moving, nearest neighbor distance is extremely important. All our simulations, all our theories, all what are DMS, all simulation only. They are so dependent on force field. We need sub-angstrom spatial resolution, and it cannot do, laser cannot do it. So what has done now, synchrotron, what happened in synchrotron, you put them around a cylindrical, some kind of a subtle and toroid actually, and you very high speed, and when it bends, and we know all electromagnetic theory, the change of velocity gives rise to electromagnetic radiation. That's where you go, and you will see all these different channels coming out. They are high intensity X-ray. High intensity X-ray allowed something. They allowed to do XAP. Before that, in SSU, they're doing XAP, but they are getting only the peak position of, say, oxygen 531 electron volt. But now you have very high intensity X-ray, and this is what happens. I'll tell you a little bit in 1 minute later. So the experiment many times, they do femtosecond laser pulse to initiate a structural change, and they send X-ray probe pulse at regular intervals. Now they needed picosecond laser. They got picosecond laser, but that was not enough. Now they have free electron laser, which is femtosecond. So they have a femtosecond pulse, which is a pump. So your pump is a laser pulse. There are other ways also doing, but there is a probe is your X-ray. It's a pump probe experiment. I told you very much from laser. How do you do now? So this is the technique. Comes from X-ray, comes from synchrotron. Then you can do fluorescence, X-ray absorption spectroscopy. But I'll tell you what transmission is the one that I'm more interested in, because fluorescence doesn't always say the X-ray absorption. And this is a beautiful thing by Shedwick, coordinated chemical train from there. Now, what is the use of XAP? I told you, first thing I learned after SSU was XAP. But this is time domain. One beautiful thing happened by following Einstein. You give a high intensity X-ray. You photoionize the electron. Electron is a wave. It goes out and it backscatters from the nearest neighbor. Now, so you do a like by reference one, which goes outside, and you get the new X-ray absorption, which is now augmented by synchrotron, by the constructive interference. Also could be destructive interference. The difference give you the positions about A. So laser excitation, and that is one thing. Send high energy X-ray pulse. Detect incident and transmission signals. Transmission signals get enhanced due to interference with scattered X-ray. Take the difference. You get the nearest neighbor structure. In disordered systems, liquids and glass, there are a lot of results now. I didn't know so much. Thanks, Ranjit, for asking me to get it done. You get radial distribution function by XAPs. Because of the availability of X-ray pulses from synchrotron, one can have a time-resolved measurement of structural change. That's amazing. So now we can see, single molecule spectroscopy uses microscope, but it uses microscope to see light moving. They didn't quite see the light moving, but they probe the light. But here you can really see, it's more powerful, but I think X-ray has its own story, own limitation. It must be damaging the system. That's what I think. Remember, Amit Reyes was doing electron diffraction talk, and Casey Hans stood and told us, but you are going to excite it anyway. So what is the proof that you are doing any reaction? Amit didn't have a single answer to that. So there are issues, but it's good to know that what is involved, synchrotron X-ray, that these days one can have a picosecond pulse of X-ray in high intensity X-ray. You hit it, you do photoelectric effect, go to the beyond the valence electron, then they spread out as a wave, get scattered, come back, they contact, and you have beautiful pictures actually that are full of air. So that's it. I don't think I'll go through anymore. What's the time now?
It's 4:38, sir.
Okay, then it's done. I think these are I just go through. I'm not telling anything. So these are top. The biological function of these two beautiful papers written by these two guys. One is how water is detected, how water role is in protein. That's amazing. That so these two PRLs were not really. The referees didn't have much to say. And the other one is calculating the two reaction, three reaction or four reaction coordinate. We calculate the force time position function. And we found the friction decreases a lot because of cross correlation, which was also this cross correlation also was here. That was what this part was to a great extent Shomok's idea. We are looking into that, but looking into one particular feature was Shomok. Okay, that's it. And these are the things I someday want to study. It as I actually have no group now, and it's a beautiful problem to study, but it's not being studied. This is a wonderful problem even to study by X-ray diffraction. This is it, and this is a beautiful poster. He gave it to me himself. I fought with him, of course, and you see the horse on the top light. Thank you very much.
Thank you, sir. Now the floor is open for questions. I don't see any in the chat box. If anyone, if you are having questions, please raise your hand so that I can ask one by one. Or you can.
There's so many people there. I thought they have all left.
No, they are here, sir. Yeah.
Hey, Shuman, ask a question. I let me understand what you understand.
Shumanda just went out, so he will join us.
Yeah. His telephone call. If not,
Sir, can I?
Yeah, Shobanda, please go ahead.
Sir, I'm Shobhan.
yeah,
I'm
bolo
Shobhan.
baba.
No. So you mentioned one very interesting thing, and you said that somebody should really think about an experiment, that these very important layers of dielectric layers around
Yeah.
proteins or any other biomolecule. Now, to really track these independent layers and their properties in terms of static properties as well as dynamic properties, what do you think, or what is your kind of suggestion that what kind of experiment one can do? Because these layers are very, very small, like 3
the layers
angstrom,
are
4
also
angstrom.
here. See, layers are
Yeah,
here.
this one. Yeah.
Layers in ions, solution of ions in water. Everywhere the layers are there. We are once one of my esteemed friend, I won't say the name abroad, said something very nice to me. I said, how come we did such a bad job, physical chemist, in solving the problems, leaving it for biologists? He said that because we are just lazy. I think what Konkon again and again told me, that first layer that he did, and that he said it is like methanol. I think dielectric constant 34, 35. Well, you know, the Somit's experiment, which actually I'm told many times that was Konkon's idea, that to put a guy outside through biochemistry, dye at different distances. You went to some 20 angstrom, no, 6 or 7 layers, and he found his bulk water.
Yeah.
But selling that did an X-ray. Shobhan, if you think sometime, I'm really telling you, I don't know whether you get the money.
Yeah.
Time domain X-ray, you should try. You know,
time domain X-ray.
Time domain X-ray. Single molecule is okay, but time domain X-ray gives much more. Of course, you need much more facilities. You need a synchrotron, but synchrotron you can go. And people see SSU people are being day and night going to synchrotron.
Yeah.
They get grant.
Yeah.
What they do, they just take X-ray diffraction. Bullshit. You should really use this X-ray. Now there are so many beautiful. Hamburg has it. argon has it. There are all these beautiful, beautiful things, you know.
Hmm.
Not a single guy in India doing. And it's good to pamper people, but I think sometimes one feels very frustrated, you know.
Yeah. Yeah. But this is a very open question. I think this is what we always observe in simulations and many other theory. But then really looking at in the experimental point of view, these layers, that becomes much more difficult, I believe. Yeah.
Yeah. Selinger wrote a feature article in JPC where he very eloquently in the introduction itself or somewhere described or work about a paragraph or so. He put the number, it is 20 angstrom. Now he did X-ray. How did he got it 20 angstrom and how did X-ray could distinguish between the layers? You will be able to figure out, but I have not been able to figure out. I still believe, that we physical chemists don't use X-ray as much as we should. You know, there's so much beautiful information and there's so much bigger audience there when you do X-ray.
Yeah. Absolutely. Absolutely.
If you just go on laser absorption, fluorescence, all these things, they're not much there. You know. These are our small community.
Yeah. True. Thank you, sir. Thank you.
Sure.
Thank you.
Okay, sir. There is one more question asked by Ashu. Ashu, would you like to ask yourself? Hello.
Yes.
Yeah,
Can I?
please.
Am I
Please,
audible?
please go ahead. Yes.
Hello, sir.
Yes.
Thank you for the nice talk. So, sir, I have a question. Like when water is between the protein layers, and you show that it has very different dielectric constant. So, what kind of dynamics are expected of such water? So, I'm.
You have, let me tell, if you have two proteins and you have water in between, these are simulations. The reason these days I'm a little bit skeptic of simulations because I think gross things are right, but detailed things I don't know. So you have, say, two protein and even in lipid, same thing, lipid bilayers and all these things. And so water when you have these kind of things, they can be either slow or fast. We found that in insulin, that it becomes considerably faster. It becomes faster by almost factor of 2 or 3 probably, if I can find that. Okay, here. Do I have that? No, I don't have. So this is the water. We did the water here in the middle of two proteins. The red ones are the hydrophobic guys. So one there here are the ones which are faster, much faster, and which was a bit surprising, but because of frustration. And the question you are asking is also a hugely important question called hydrophobic force law, HFL. And that when electron microscopes study that when two surfaces are close together, is there attraction or repulsion? And that is really tough, hydrophobic force law, HFL. And we have a paper, 2014 Soikot, on that hydrophobic force law, and we had a lot of the references we'll find there. Very nice experiments have been done on two paraffin surfaces with a cantilever. You put them in a cell and bring them together. You allow the water to escape through another aperture, but you can bring them together, and then you have to cantilever to two plates, and you will be able to see measure the force between the plates. That has been done. But what is a physical chemist would like to know, or chemical physicist or even physicist, is that what is the structure of water in between? And that we know nothing other than simulations. When I was young, I would say other way around. You said, okay, you cannot you cannot do. It's great that we can do it in simulation. You cannot do in experiment. But over the years, you do need these experimental measurements. I think somehow X-ray will be able to do these kind of things, but I have no clue how, what can. X-ray's problem is that with all its greatness, it's a high intensity X-ray. In order to get local structure in a disordered system like liquid and glass, you need high intensity X-ray. But high intensity X-ray is dumping a lot of energy, right?
Yes.
So, in water, then you have to focus, but the resolution is sub-angstrom. If I can focus on one water, or I can dope it with iron, and then if I can, there is a excitation for the K electron, which are core electrons. I can excite it to outside, but then the signal will be very, very low, because yes, electron will become a wave and scatter back, but then and I will detect the enhanced X-ray scattering, which is XAP, time domain XAP. But that will be, it's an amazing experimental technique. Just wait for, I think, a few years, you get Nobel Prize. They have given Nobel Prize to everything. They have given Manfred Eigen for classophotolysis, and they have given to Dudley Hertzberg. They have given to Youngly Dudley Hertzberg. They have given to as well. They have given 2, 3 in between for laser. Townsend, they gave to Townsend. They gave to Bloembergen. They gave to then all the electron microscopes. Each of them got a Nobel Prize, each technique. Then they have given to cinemolecular spectroscopy and electron diffraction, I think last year. Now time of the X-ray. But what makes me a little sad, the reason I'm willing to talk, that students should know about this technique, because so much jobs, huge amount of jobs in these things, And here our students and theory students are not getting job, but then I get so many, I have 3 PhD positions, I have these positions, I have postdoc, but all this kind of big national lab or international labs.
Sir, my next question is relating, nowadays people talk about isolated water in salt kind of solution. They're like 21 molal or some kind of salt solution are used for batteries. So they're like, with the iron, there is single water, and nearby there is long iron chain. So these isolated water has like water splitting window is enhanced, like it doesn't split at 1.23 volt. But it acts like a supporting solution for water and salt kind of batteries.
Yeah, you have to look into this. We wrote a paper with Ananda Biman Bhattacharya, because he does some battery work. So the problem that we did is very similar to, he was interested to know into one of those solids, maybe perovskite or something else, whether lithium will go in or zinc will go in. And then so lithium is interestingly, people don't know, lithium plus and zinc 2 plus are 1.89 and 1.91 angstrom, same size. But zinc has 2 charges, but they are little far.
Yes, sir.
Lithium has 1 charge, but they are very close. So this competition is really interesting. Who goes? That we figured out that who goes, and if this guy goes with a few water, it turned out that's what other people also saying. Now, in your question, you have the ions. Okay. In biology also, many things are done by single water.
Yes,
It's
sir.
amazing. One water molecule does all the function. Now, so it will be interesting if, let us see that I have lithium chloride or something like that, or zinc chloride, and I have counter ions, or all the cation and anion. Now, cation and anion now together solvating my water, right? And so that would be interesting structure to find out. I can tell one of the students to do it. Okay, put one water molecule. That would be a lot of fun to see what really happens. That is the melt, but people have studied melt, but people have not studied melt with such a low concentration of water. It's the other way around, we think. It'll be interesting.
Thank you, sir.
I think Snehashis has raised hand. Snehashis, can you please go ahead and ask your question?
Hello, sir. Is it
Yeah,
audible?
how are
right?
you?
Yes,
Yeah,
yes.
I'm good. I'm good, sir. I'm good.
You are having a lot of sushi.
I was having, but now I have reached home, so I'm
Oh,
now
come
in
back.
Patna.
Oh,
Yeah, I
okay.
have come back.
Uh,
Sir, the talk is great, and as usual. So I have a question which might sound a little bit silly. That this spatial resolution and time resolution. That normally when we do two things, we think separately. But is it possible that the spatial resolution and time scale, or spatial scale and time scales, are somehow connected and correlated?
yeah. I think in most of the cases they are connected. In enzyme
Yeah, okay.
kinetics, everywhere we need the two things together. That's what I was saying. That you have get no structure in one way, and actually very rudiment. I think at a level of technique is a great technique, but the level of information, laser all these things have been by and large. You have to search for problems.
No. So maybe my questions, I have not been able to put it correctly. So what I'm trying to tell is, let's say you study a very small system, okay, and of course it is a small collection of a large system. And when you do that, do you get the time scales which you are getting from experiment, which is a
No.
large system,
No.
is same, or there is
No,
a good
because-
correlation between that?
No, there is always a danger
Mm.
of collective thing. Like, for example, the study of water in a sphere or in a nano slit. So if
Yes.
you confine this here, then we found, I think, to get the bulk dielectric constant, you have to go some 10,000 water molecules.
Water
Very
molecules.
big water molecules. Now, but there are single particle properties that arrive at bulk properties very quickly, but not the collective properties. So what you are saying, can my collective small system of 1,000 or 2,000 can capture the collective properties? I said no. That you will have, because collective properties are actually, that in long wavelength, k equal to 0, k is the wave vector, k equal to 0 limit. And
Yes,
so they are
collective limit.
really populated. Things are really populated. That's why viscosity and friction dependence is actually fractional. Is one of the way of that very
Right.
short time you don't get the bulk.
And also dielectric relaxation, I think to study the dielectric relaxation, you need to have much larger systems than normally
nobody
to study
has
the
done
diffusions.
that. Yeah, absolutely. Yes. I see, this famous Gelm science paper. There are two dangers. Middle region becomes okay, but we cannot study the middle region. So we are studying the dielectric constant. There's a science paper of Gelm. And I think we wrote a science on a nanoliter on that. And what we found is that this interpretation of the dielectric constant is because it is in series of capacitance. So 1 over epsilon measured by putting charges on the plates, positive or negative charges on the two plates, is sum over 1 over dielectric constant. So the dielectric constant that is near the liquid that is near the surface, its dielectric constant is much less and it dominates. So one dielectric constant becomes 2. So it becomes 5. 1 over 80, that's 0.01 or 0.012. So there are a lot of interesting things you have to look into. I think one good thing that working with the experimentalist and the comparison between theory and experiment teaches you. What are the questions? There are millions of questions, but you cannot go on asking every question. You need to ask the question which can be verified in experiments and which can throw light on something, a correct light. It has to be, I can go on doing things which are actually meaningless.
Mhm.
Even those who are doing quantum calculations, materials, I think, I often see them giving a talk without they give a very broad phenomenon thing, but they do not talk of the specific thing.
Mhm.
And specific things will always be spectroscopy or this kind of diffraction. It's a very good question.
Thank you, sir.
Okay, next question is asked by Shomajit. Shomajit, would you like to ask your question very briefly? Shomajit, are you there? Okay, maybe I will read. Sir, the dielectric constant is often taken to be 4 inside the protein environment. Even in quantum mechanical calculation, dielectric constant 4.0 is sometimes used to accommodate the effect of the protein environment. How much correct value it should be taken? What is the correct value?
So people take between 2 and 4. I know most of the time it is 3.
Okay.
Okay. Now, within the protein, you have very few water. So what are the things? One should read the Lellinger, the famous writing on proteins. He didn't write. He just did Corey, Pauling and Corey. They are Corey and Pauling quoted just. After 1 or 2 lines, he just within quotation mark, he put Pauling wrote. Pauling wrote a very interesting thing. He said, look, you should think of alpha helices as liquids and beta sheets as crystal. Now, if it is a crystal, it has dielectric constant, of course, only the electronic dielectric constant. There's no rotation, orientation. But helices do contribute some because it fluctuates. Dielectric constant is the mean square dipole moment fluctuation. Every guy should know that. It is M square, capital M. M is sum over all the dipole. Square of that. And then take the average. That gives you the dielectric constant in a sphere by Clausius Poisson equation. Otherwise, there are other equations, but very similar thing. Now, M square is very small because there is very little water, and then beta sheets, they have dipole moment, but they are not fluctuating. And that's why it is 2 and 3. And that's what the main result of main result of Shomoksha and that it's a water from outside goes inside, but that if you have charge inside, then you are looking into what they do. They screening of charges. So they say you have a alpha helix, which is a dipole moment. Remember the peptide dipole bond. A peptide dipole bond. A peptide bond has a dipole moment of 3.45, which is huge. People forget that peptide bond is highly dipolar because C double bond in H. So they had to screen. And that screening is done by taking the dielectric constant between 2 and 4. That's empirical, but that works. That's what I said. Many things we don't understand. And I once reviewed a series of papers in Biophysical Journal by a European author. Starts with S. I forgot the name. Protein dialectic. You can easily do the Google. Protein dialectric constant. Very, very nice bit of detail work. They also got that small thing because you don't have orientation too much.
Thank you, sir. The last question is asked by Oinda. Oinda, can you please ask, sir?
Okay.
Yeah.
Am I audible?
Yes, you are.
Okay. Wonderful lecture, Professor Bagchi. So, I mean, generally coming from a more of a kind of a quantum material kind of perspective. So, one thing I've found that it has become possible now from an electronic structure viewpoint, from a density functional theory base, to look at surfaces, solids, materials, but which are more pristine type. But the physical chemistry in general lies in the defects, in the disorder. Somehow that part, I would say from my community, there are no, it's not that there is a computational limitation. Computational limitations are always there, but there is serious lack of understanding of the models. And
Yeah,
and the,
I think we discussed it once little bit long
yes, sir.
time ago. Now, what I'm saying is, if you see, you first look into when you do theory. It's good to do theory. I'm not saying no, but what kind of experimental verification you can have? If there are defects, like one figure I showed was from excitons on chalcogenite surfaces. I think very fast one probably. These are all stolen from internet. If I can find it, that Baba. No, I don't think I'll be able to find it. No, I don't. But what happened that is this sum of Lorentzians and your so you need to have some kind of a absorption experiments done. Okay, and you need to get the energy levels. I agree that defects and all these things, but I believe you have now quantum chemical methods to get some quantitative answer. And then you do the theory. If it is a pristine condition, then yes, we do theory for ourselves, theoreticians theory, but an experimental will not be interested at all. It reminded me of a great Gandhi movie, that when everybody was talking all these things, saying that we need Swaraj, we need this, we need freedom, in a big congregation. That was the first time Gandhi was going to speak. Abdul Kalam Azad and Vallabhbhai Patel put it there. I think Abdul. So Gandhi said something great thing. He said everybody was leaving because nobody knew him, but he started speaking that, gentlemen, India is 7 large villages. And here what we do, what we talk, lecture we do, makes no difference to Indians. It might get a few lines in progressive newspapers and we read ourselves. It has no impact. I think my point is that I'm not blaming theoreticians. I think experimentalists are equally to blame. Like I have tried much in SSU to collaborate with them, that you should get somebody to do some absorption experiments with defect. Then if you can get things and your quantum calculation you can do, if you can tell them a little bit more about the nature of the electronic state or excited state, and whether people do these things for certain use in mind, right? And so you can probably ask the right question. I wrote a blog, you remember, Ryan, that we didn't read it probably. I said that much of the work that we do from India get published in good journals, 1 or 2 citation it gets, but really no impact. And one of the reasons is that, but if you do something, people always, interesting people will always notice. I think we are not asking the right questions. Of course, X-ray and single molecule, we don't have a single molecule spectroscopy unless some on Indo and Sohan probably doing some, but even then they are in spectroscopic sense, not material science sense. Then you also have I would imagine that you have to go ahead, do the calculation, you have to get somebody, do the measurement. Then you see when you write, people will notice it.
Thank you, sir. Thank you. Wonderful.
Okay, so with that, I think there is no more questions. So let us thank Mr. Bagchi once again, and I request Shumanda to take over.
Suman went out.
No,
No,
I
he
just
came
went
back.
out for 5 minutes.
Came
So, yeah, Thank you again. Thanks, Rajiv. And I thank Professor Bagchi for a very insightful, first of all, historical perspective, as well as these insights about connecting the importance of bridging the gap between theory and experiment. Somebody.
That's
Okay.
very interesting. Yeah. That's not from me.
Okay. Yeah. So thank you, Professor Bagchi, and thanks everybody. Before we disperse, we'll have couple of important announcements. So we have two very big conferences coming up. One would be for the Society of Physical Chemistry conference that would be organized by IIT Bombay Chemistry Department. And for that, I request Professor Anindo Dotto, who is one of the conveners, to please make the announcement about the SOFIC conference.
Thank you, Shuman. I will share my screen and I'll take just maybe 1 minute.
No, take more.
Yeah. I hope you can see my screen.
Yes.
This is the website of the second SOFIC conference. Society for Physical Chemistry was formed last year, and a lot of people who are present here are very active in the society. Many of the speakers, Professor Bagchi has really been the reason why this society has formed. Last year, we had our conference in IIT Kanpur. This year, we have it in IIT Bombay. The dates, as I mentioned on the website, are 22nd to 25th October. This
Can you make the dates, that thing little bolder?
Yes, but
make it a little bolder.
I can zoom in. Here it is.
Even then, they are also make it bold. Bold is always very
Yes, we'll do that. This is the organizing committee. As you can see, this is really a SOFIC meeting. So the behind the scene, everything is done by the organizing committee or SOFIC office members. We have a good national advisory committee, and the local organizing committee comprises faculty members from IIT Bombay, DRC, TIFR, and DAE University of Mumbai Center for Education on Basic Sciences. We already have confirmations from a number of very eminent physical chemists across the country, across disciplines, and across age groups. The talk we heard today from Professor Bagchi was all about experiment and theory working hand in hand in synergy. So if we just scroll through the speaker list, I hope one can see the same synergy in action. So the announcement in this talk is very appropriate because the talk sort of sets the background of why we are doing this symposium in the first place. As you can see, all the areas of physical chemistry are represented very well. The purpose of this meeting is to not only hear talks, but also to talk to each other, build collaborations for students to give them new ideas and so on and so forth. Other than talks, since we want maximum amount of involvement of senior researchers as well, we have this very unique concept of faculty posters, and one very eminent photograph is missing in the list as of now, but we'll make amends.
Hey, where is my photograph?
That is what I was saying nicely. So,
not too nice. You missed my picture.
Professor Bagchi, Professor Bagchi also presents a poster here.
Last time.
Yes, this time also you are going to give it. And many of us will present posters. So as you understand, within a span of 3.5 days, it's not possible to have too many talks. Otherwise, we get saturated. Posters are a good workaround. And we have eminent researcher chairpersons as well. But this is a chairperson. This is something that you perhaps don't enjoy as much as posters. This is a new society. We do not really have any corpus at the moment. So we will need to raise the expenses largely from registration fee. And also I'll talk about sponsorship in a moment. So registration fees are 18,000 rupees plus GST for faculty and scientists who need accommodation, 9,000 plus GST for those who don't. So in case you have a place to stay in Mumbai, you don't want to stay in our guest house, you pay half the registration fees. And for students and postdoctoral fellows, the fees are 8,000, including shared accommodation in hostels, 7,000 without. The schedule is not updated yet, but you remember the dates, 22nd to 25th October. And we have very generous sponsorship from IIT Bombay to start with. from the Institute of Eminence program. This is where the venue is going to be, Victor Menezes Convention Center in our institute. The website went online only yesterday, so it is very much a work in progress, but please look for SOFIC 2024 and we'll keep updating it. We do have a list of about 500 active physical chemists of the country. So we plan to write to all of them very soon. And once we do that, hopefully everybody will get a complete information. So this was meant to be at least a starter. So thank you very much, Shuman, for getting this started.
Yes, thank you, Anindada. We are all looking forward to this meeting. Now that there will be announcement of another meeting, which is for this particular platform, SMCB. There will be one in-person seminar, and to announce that, I request Professor Rajiv Bishwas from IIT Tirupati. Rajiv, please.
Thank you, Shumanda. So let me share my screen also. I think is it visible?
Yes.
Okay, so yeah, this SMCB platform was, it has been, as Shumanda already spoke during the beginning of this today's session, it was the brainchild of Professor Bagchi. So it was started when we all were facing that COVID-19 pandemic peak situation. So somewhere around January 2021. And the first conference online mode we had in February 2021. So this will be the first in-person meeting, and we are at the Department of Chemistry of IIT Tirupati are really fortunate to host the first in-person meeting of SMCB 2024. The date are mentioned here 16 to 19, but hopefully we'll be able to make 3 days so that we'll work it out. And this is the flyer for this, and myself and one of my colleagues, Dr. Devashish Mondal, are the conveners, and we have the organizers, where we have Professor Ranjit Bishwas and Professor Suman Chakraborty, my co-coordinators of this SMCB seminar series platform, and we have a couple of other colleagues, Arun K. Manna from IIT Tirupati, Rakesh from IISc Tirupati, and Anand Srivastava from IISc Bangalore. And of course, there will be many more people involved over the course of time as we make progress. We don't have right now the updated data like Anindada showed, so we are working on it. And this is kind of a fast announcement, and I request all the interested students and other faculty members to look and browse through this website and look for the updated announcement, which will be sent to SMCB Google groups in due course of time. So thank you, Shumata.
Okay, thank you, Rajiv. So another exciting conference to look forward to. So with that, we come to the end of today's proceedings. Once again, I thank you all for being here till the end, and the recording will be made.
Hey, so good that I gave the talk, because they Anindo and Rajiv could make the announcement.
Yes.
Yeah.
Absolutely.
Yeah, we managed to get a lot of attendees because of this special attraction. So it's a good point to now announce this. Okay, so yeah, thank you all. And the recording of the talk will be made available in our YouTube channel as usual, and future events will be announced soon in our mailing list. So with that, I close today's event, and thank you, everybody. Take care.
Thank you.
Bye.
Bye-bye.
Thank you.
Bye,
Thank you.
everybody.
Bye. Bye.
Bye.